Welcome to the deep dive. Today we're diving into an image you see everywhere on t-shirts and pop culture. It's a symbol of life itself, the iconic DNA double helix. But do you really know what it represents? We're taking a deep dive into the molecular basis of inheritance. We'll explore how scientists uncovered the DNA as life's master blueprint, how it copies itself with astonishing accuracy, how it's meticulously packaged, and how understanding it has completely transformed biology and medicine.
Right. Our mission today is really to unpack this foundational chapter in biology. We'll trace the incredible journey from those initial scientific inquiries. It's a real scientific detective story, actually all the way to the cutting-edge technologies that now let us read and even, edit life's most fundamental instructions. Prepare for some truly surprising insights and, well, profound real-world implications.
And the sources for this deep dive are excerpts from Campbell Biology in Focus, 3rd edition. So let's unpack this. Okay, so before DNA became like a household name, the question of what actually carried genetic information was one of the biggest mysteries in biology. For a long time, proteins were the prime suspects. And you can see why, right?
Absolutely. Proteins were known for their incredible diversity, their specialized functions. It just seemed necessary for something as complex as heredity.
Yeah, how could some simple molecule carry all that information?
And what's fascinating here is that nucleic acids, like DNA, they seemed far too simple chemically, just 4 repeating units. How could that account for all the inherited traits we see? But that view began to shift dramatically with experiments using much simpler biological systems. Bacteria and the viruses that infect them, bacteriophages.
Which raises a really important question. How do you convince the entire scientific world of something so fundamental, something that completely redefines our understanding of life itself?
Yeah, that's a huge hurdle.
The first really big clue came way back in 1928 from a British medical officer, Frederick Griffith. He was actually trying to develop a pneumonia vaccine. He worked with 2 strains of Streptococcus pneumonia, a disease-causing S strain, which had this protective capsule, and a harmless R strain, which didn't.
Okay.
And here's where it got really puzzling. Griffith found that if he heat-killed the deadly S cells, so they were dead, and then mixed their remains with living harmless R cells, some of those living R cells actually became deadly.
Yeah.
And even more astonishing, this new deadly trait was inherited by all their descendants.
Wow. So something was passed from the dead S cells to the live R cells.
Exactly.
Griffith called this transformation basically a heritable change. The cell's genetic instructions, its genotype, were permanently altered, which then showed up as a new characteristic, its phenotype, like being able to cause disease. But the identity of this transforming substance, well, it remained a mystery to him. Later work by Oswald Avery and his team pointed strongly to DNA.
But people were still skeptical.
Oh, very skeptical. Proteins were still the favored candidate for many. It took more evidence.
Right. So fast forward to 1952. Alfred Hershey and Martha Chase conducted this landmark experiment. Really powerful evidence. They used bacteriophages, or phages, these viruses that infect bacteria.
And phages are incredibly simple, basically just DNA wrapped in a protein coat. That simplicity is key.
Scientists knew these phages could somehow reprogram bacterial cells to make more viruses. But the crucial question was, which part did the reprogramming? The protein coat or the DNA inside?
And what's truly elegant is their experimental design. They used radioactive labels. Smart stuff.
Yeah, they tagged the protein with radioactive sulfur-35 because protein has sulfur, but DNA mostly doesn't. And they tagged the DNA with radioactive phosphorus-32 because nearly all the phage's phosphorus is in its DNA, not the protein.
Okay, so distinct labels for each part.
Exactly. They infected separate batches of bacteria with each type of labeled phage. Then, and this is clever, they agitated the mixtures in a blender to knock the phages off the outside of the bacteria.
Right, separating them.
Then they spun them in a centrifuge, so the heavier bacteria formed a pellet at the bottom.
And the results.
Incredibly clear. When the protein was labeled, the radioactivity stayed mostly outside the bacterial cells, in the liquid supernatant.
Okay.
But when the DNA was labeled, the radioactivity was found inside the bacterial cells, in the pellet.
Ah, so the DNA went in.
Yes, and the clincher. Those cells containing the radioactive phage DNA went on to produce new phages that also contain that radioactive phosphorus.
That's pretty decisive. It showed that the phage DNA, not the protein, entered the cells and carried the genetic information needed to make new viruses.
Absolutely. This experiment was a real game changer. It finally convinced most of the scientific community. DNA was the hereditary material.
But there was still another piece of the puzzle missing, right? About the composition.
Yes, exactly. By 1950, biochemist Erwin Chargaff had added crucial data, Chargaff's rules. He analyzed the chemical makeup of DNA from lots of different organisms.
And found some strange patterns.
Two peculiar regularities. First, the DNA-base composition varied quite a bit between species. Like sea urchin DNA has about 32.8% adenine, while E. coli only has 24.7%.
Which actually made DNA seem more like genetic material because that diversity could account for species differences.
Right. It wasn't too simple after all.
And the second rule.
This was really surprising. In every species he studied, the percentage of adenine A bases always approximately equaled the percentage of thymine T bases. And similarly, the percentage of guanine G bases always approximately equal the percentage of cytosine C bases. A matches D, G matches.
The amounts were always balanced, but nobody knew why.
Exactly. The reason remained a mystery until the structure itself was figured out. So you have Griffith's transformation, Hershey and Chase proving DNA carries the info, Chargaff showing these ATGC rules. The stage was set. And then April 1953, James Watson and Francis-Crick, using all this prior work crucially, including X-ray diffraction images from Rosalind Franklin and Maurice Wilkins.
Those images were key. Franklin's photo 51 especially.
They unveiled their revolutionary molecular model, the double helix.
And Watson and Crick it was this double helix, two sugar phosphate backbones winding around the outside, like a twisted ladder. But importantly, the two backbones run in opposite directions. They're antiparallel. One runs 5' to 3', the other 3' to 5'.
Like a divided highway. That detail turns out to be critical for copying.
Absolutely. And the nitrogenous bases A, T, G, and C, they project inwards, forming the rungs of that ladder.
They realized, looking at Franklin's data showing a uniform width, that a purine, the bigger two-ring bases A or G, must always pair with a pyrimidine, the smaller single-ring bases C or T.
Keeps the width constant. Purine would be too wide, pyrimidine too narrow?
Exactly. And the specific pairing, A with T and G with C, was dictated by how hydrogen bonds could form between them.
Right. Adenine forms 2 hydrogen bonds specifically with thymine, and guanine forms 3 hydrogen bonds specifically with cytosine. It's chemically specific.
And this elegant base pairing explained Chargaff's rules perfectly. If A always pairs with T, their amounts must be equal. Same for G and C.
It all clicked into place.
So what does this all mean? The structure wasn't just beautiful. It immediately suggested how genetic material could be copied.
As Watson and Crick famously understated in their paper, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material.
Such a classic line. The idea is simple yet profound. Each strand acts as a template.
Right. You separate the strands, and each old strand dictates the sequence of a new complementary strand being built alongside it. A pairs with T, G pairs with C. This leads to the semiconservative model of replication. Each new DNA molecule has one old parental strand and one brand new strand.
And that model was confirmed experimentally just a few years later by Meselson-Stahl using heavy nitrogen isotopes. Really elegant work.
Yeah, classic experiment.
Okay, so the cell needs to copy its DNA. Let's think about the scale. A single human cell has about 6 billion nucleotide pairs.
That's a staggering amount of information, like copying 1,400 textbooks.
And it has to do it in just a few hours during cell division. With incredible accuracy, maybe one error per 10 billion nucleotides. How is that even possible?
It requires what we call a DNA replication machine, a whole complex of maybe a dozen or more enzymes and other proteins working together in a highly coordinated way.
Where does it start?
Replication begins at specific sites called origins of replication. Bacteria usually have just one origin on their circular chromosome. It opens up a replication bubble that grows in both directions.
But eukaryotes, with our huge linear chromosomes.
We have hundreds or even thousands of origins per chromosome. This allows multiple bubbles to form and eventually merge, which dramatically speeds up the whole copying process.
Makes sense. And at each end of a bubble is a replication fork.
Exactly. A Y-shaped region where the parental DNA strands are actively being unwound, like a zipper opening up.
So who's doing the unzipping?
Several key players. Helicases are the enzymes that untwist and separate the strands. Then, single-strand binding proteins latch onto the separated strands to prevent them from immediately reappearing, like holding the zipper open.
Got it.
And as you unwind, you create tension and supercoiling ahead of the fork. Topoisomerase relieves that strain by cutting, swiveling, and rejoining the DNA strands.
Wow, quite the molecular machinery. So Once the strands are separated, how is the new DNA built?
The main enzymes are called DNA polymerases. They add nucleotides one by one to the new growing strand, following the template.
But you mentioned a catch.
Yes, DNA polymerases can't start a new chain from scratch. They need something to add on to. They can only add nucleotides to the 3' end of an existing strand.
So how does it get started?
That's where Primus comes in. Primus makes a short RNA segment, maybe 5-10 nucleotides long, called a primer. This provides the free 3' end that DNA polymerase needs to get going.
An RNA primer to start DNA synthesis. Interesting.
Yeah. Then the main DNA polymerase, often called DNA polymerase III in E. coli, takes over, adding DNA nucleotides to that primer's 3' end, using the parental strand as a template.
And you mentioned DNA strands are antiparallel, and polymerase only adds to the 3' end. Does that cause issues?
It does. It means the two new strands have to be synthesized differently because they are growing in opposite directions relative to the moving fork.
How so?
Along one template strand, the one oriented 3 to 5 foot towards the fork, the polymerase can synthesize the new strand, called the leading strand, continuously. It just follows the helicase as it unzips. Nice and smooth, 5 to 3 foot.
Okay, one smooth piece.
But the other template strand runs 5 to 3 foot towards the fork. Since polymerase must synthesize 5 to 3 foot, it has to work away from the replication fork on this strand.
So it has to keep starting over.
Exactly. This strand, the lagging strand, is synthesized discontinuously in a series of short segments called Okazaki fragments.
Okazaki fragments. How short are they?
About 1,000 to 2,000 nucleotides in bacteria, maybe 100 to 200 in eukaryotes. Each fragment requires its own RNA primer laid down by Primus.
So the lagging strand is built piece by piece.
Right. After DNA pol 3 makes a fragment, another polymerase, DNA polymerase I, comes in and replaces the RNA nucleotides of the primer with DNA nucleotides.
Okay, swapping RNA for DNA.
And then finally, the enzyme DNA ligase acts like molecular glue. It joins the sugar phosphate backbones of all the Okazaki fragments together, creating one continuous strand.
It's incredible. All these enzymes working together at the fork, coordinating leading and lagging strand synthesis, often visualized with the trombone model where the lagging strand loops out.
It's a beautifully complex and efficient machine.
But you said even with all this, the initial error rate is maybe 1 in 100,000. How do we get to 1 in 10 billion? That accuracy is mind-blowing.
It's largely due to proofreading and repair mechanisms. Many DNA polymerases actually proofread each nucleotide as they add it.
Like hitting backspace.
Exactly like that. If it adds the wrong nucleotide, it can immediately remove it and try again. That catches most errors.
Wow.
And beyond that, other enzymes perform mismatch repair, scanning the newly made DNA and correcting errors that slipped through the initial proofreading.
And these repair systems are critical for health.
Absolutely vital. Defects in mismatch repair genes, for example, are linked to hereditary forms of colon cancer. Our cells are also constantly repairing damage to existing DNA caused by environmental factors.
Like UV light from the sun.
Yes, UV light can cause adjacent thymine bases to link together, forming thymine dimers, which distort the DNA helix.
How's that fixed?
A system called nucleotide excision repair handles it. A nuclease enzyme cuts out the damaged segment. Then DNA polymerase fills the gap using the undamaged strand as a template, and DNA ligase seals it up.
So the undamaged strand provides the backup copy.
Precisely. And the importance is starkly clear in diseases like xeroderma pigmentosum or XP. People with XP have defects in these repair enzymes.
And they're hypersensitive to sunlight.
Extremely. They often develop skin cancer at a very young age because their cells just can't fix the UV damage effectively.
It really highlights how crucial these repair mechanisms are. But if replication and repair are so accurate, where do mutations, the fuel for evolution, actually come from?
Well, even with all these systems, very rarely, a mistake does slip through and isn't caught. Once that mismatched nucleotide pair is replicated in the next round of cell division, the change becomes permanent in that lineage. That's a mutation.
So it's a numbers game. Errors are rare, but DNA is huge and cells divide a lot.
Exactly. And while most mutations are harmful or neutral, that very small percentage that happens to be beneficial provides the raw material for natural selection. It's this balance between high fidelity and a low mutation rate that drives evolution.
Okay, one last replication challenge. What about the ends of our linear chromosomes? You mentioned polymerase needs a primer and can only add to a 3' end.
Right. This creates an end replication problem. When the final RNA primer on the very end of the lagging strand is removed, there's no way to fill that gap with DNA because there's no 3-foot end to add onto.
So the chromosome would get shorter with every replication.
It would. And it does, slightly. But eukaryotes have evolved protective caps called telomeres at the ends of their chromosomes.
What are telomeres?
They're special non-coding sequences, just repetitive DNA in humans. It's TTG repeated hundreds or thousands of times. They act as a buffer zone.
Protecting the actual genes from being eroded away.
Exactly. The telomeres get shorter with each cell division, but the genes stay intact, at least for a while.
And this shortening is linked to aging.
It's proposed to play a role, yes. As telomeres get critically short in somatic cells, it can trigger the cell to stop dividing or even die, part of the aging process in tissues.
But not in cells that need to divide indefinitely, like germ cells.
Correct. In germ cells, which produce sperm and eggs, an enzyme called telomerase is active. Telomerase actually adds length back onto the telomeres, restoring them.
So it prevents the loss of genetic information across generations.
Precisely. Interestingly, telomerase is usually inactive in most of our body somatic cells, but in many cancer cells, it gets reactivated.
Allowing them to divide indefinitely, contributing to their immortality.
That seems to be part of it, which of course makes telomerase a potential target for cancer therapies.
Fascinating. Okay, so we've copied the DNA. Now, how do you pack it all in? You said 4 centimeters of DNA per chromosome into a nucleus thousands of times smaller.
Yeah, it's an incredible feat of packaging. It involves an elaborate multi-level system of coiling and folding. The whole complex of DNA and associated protein is called chromatin.
How does it work in bacteria first? They don't have a nucleus.
Right. In bacteria, their circular DNA molecule is supercoiled, twisted up on itself, and packed with proteins into a dense region called the nucleoid. But it's not enclosed by a membrane.
Okay, eukaryotes. Much more complex.
Much more complex. The first level involves proteins called histones. These are small proteins with a lot of positive charge, which allows them to bind tightly to the negatively charged DNA backbone.
Like opposite charges attracting.
Exactly. DNA winds about twice around a core of 8 histone proteins, forming a structure that looks like a bead called a nucleosome.
Okay, a nucleus and bead.
And the DNA between these beads is called linker DNA. Under an electron microscope, this basic level of packing looks like beads on a string. This is the 10-nanometer fiber.
And these beads aren't just structural.
No. The histone proteins have tails that stick out, and modifications to these tails play a huge role in regulating which genes are turned on or off. So packing is linked to gene control.
Wow. So what's the next level of packing?
The 10-nanometer beads on a string, fiber coils and folds further. Interactions between the histone tails and adjacent nucleosomes cause it to form a thicker 30-nanometer fiber.
Getting more compact.
Then, this 30-nanometer fiber forms large looped domains, which are attached to a protein scaffold inside the nucleus. This creates a 300-nanometer fiber.
Looped domains. Like organizing the string into manageable bunches.
Kind of, yeah. And finally, during cell division, especially in metaphase, these looped domains coil and fold even more tightly, producing the highly condensed, characteristic X shape of a metaphase chromosome that you can actually see with a light microscope.
And this packing is precise.
Very precise and specific, ensuring genes end up in consistent locations.
Is all the chromatin packed the same way all the time?
No, it's dynamic. In interphase cells, when genes are being actively used, most chromatin is less compacted. We call this euchromatin. Its looser structure makes the DNA accessible for the machinery of transcription.
So euchromatin is working DNA.
Generally, yes. But some regions, like the centromeres and telomeres and other sections that aren't actively being transcribed, remain highly condensed even during interphase. This tightly packed, generally inactive form is called heterochromatin.
Euchromatin is open for business, heterochromatin is closed.
That's a good way to think about it. And the cell is constantly remodeling chromatin, loosening or tightening it as needed.
Incredible complexity.
Oh.
Okay, so understanding DNA structure and replication wasn't just about understanding life, it actually opened the door to manipulating it, right? Genetic engineering.
Absolutely. The discovery of the structure and how it works was the key that unlocked the ability to directly manipulate genes for practical purposes.
And the fundamental property that makes it all possible is?
Nucleic acid hybridization, that basic rule. A pairs with T, G pairs with C. The ability of one strand of DNA or RNA to find and bind specifically to its complementary sequence on another strand, that underlies almost everything in genetic engineering.
Like molecular Velcro, almost.
A very specific kind of Velcro, yes.
So one of the basic techniques is DNA cloning or gene cloning, making copies of genes.
Right. It allows scientists to make many identical copies of a particular gene or DNA segment, often using bacteria, especially E. coli, as tiny factories.
How does that work with bacteria?
Bacteria have their main chromosome, but they also often have small circular DNA molecules called plasmids that replicate independently. Scientists can use these plasmids as tools.
Okay.
You take a plasmid and you insert the foreign DNA you're interested in, say, a human gene, into it. This creates a recombinant DNA molecule, DNA from two different sources joined together.
Recombinant DNA. Got it.
Then you introduce this recombinant plasmid into a bacterial cell. Now you have a recombinant bacterium.
And when that bacterium divides?
It copies the plasmid along with its own DNA. So as the bacterium reproduces, you get a clone, a large population of genetically identical cells, all carrying and replicating that foreign DNA you inserted. The plasmid here acts as a cloning vector, a vehicle to carry the DNA into the host cell and get it copied.
So what's the point? Why make millions of copies of a gene in bacteria?
Two main reasons. First, to amplify the gene, just make lots of copies of it so you have enough to study its sequence or function of research material. Second, you can use the bacteria to produce the protein product encoded by that gene. If you put a human gene into bacteria under the right controls, the bacteria will transcribe and translate it, making the human protein.
Like using bacteria to make insulin.
Exactly. That was one of the first major successes. Human growth hormone, clot-busting proteins for heart attacks, many medically useful proteins are now produced this way. Or you can transfer cloned genes into plants, maybe to give them pest resistance.
So how do you cut and paste the DNA? You need molecular scissors, right?
Precisely. We use restriction enzymes, or restriction endonucleases. These are enzymes originally found in bacteria that recognize and cut DNA molecules only at very specific short nucleotide sequences called restriction sites.
So they cut predictably at certain spots?
Yes, very specific spots. Bacteria use them as a defense against invading viruses. They chop up the viral DNA.
Clever. And how do they cut?
Many of them make staggered cuts. They don't cut straight across both strands. They cut in a way that leaves short, single-stranded overhangs at the cut ends. These are called sticky ends.
Sticky ends. Why sticky?
Because they're single-stranded and complementary to the sticky end produced by the same restriction enzyme on any other piece of DNA. So if you cut your gene of interest and your plasmid vector with the same enzyme, their sticky ends can base pair with each other. They'll stick together through hydrogen bonds.
Allowing you to join different pieces together.
Exactly. Then you add DNA Lagos, the same enzyme that joins Okazaki fragments, and it permanently seals the backbone, creating a stable recombinant DNA molecule.
Amazing toolkit. Cut with restriction enzymes, paste with Lagos.
Pretty much. And to check if your cloning worked, or to analyze the DNA fragments, scientists often use gel electrophoresis.
How does that work?
It's a technique that separates DNA fragments based on their length. You load the DNA into wells in a gel matrix and apply an electric current. Since DNA is negatively charged, it moves towards the positive electrode.
And shorter fragments move faster.
Exactly. Shorter fragments wiggle through the gel pores more easily and travel farther than longer fragments. So it sorts the fragments by size, creating a pattern of bands you can visualize.
Okay, but what if you start with only a tiny amount of DNA, like from a crime scene or a fossil, not enough to clone directly?
That's where the polymerase chain reaction, or PCR, comes in. It's an incredibly powerful technique for amplifying a specific segment of DNA exponentially.
Making billions of copies from almost nothing.
Pretty much. You can start with a single molecule or DNA from a single cell, or even degraded DNA, like from that woolly mammoth example, and make billions of copies of your target sequence in just a few hours in a test tube.
How does PCR work?
It uses repeated cycles, usually 20 to 30 cycles of 3 steps. 1. Denaturation. Heat the DNA briefly, like 95 degrees C, to separate the two strands.
Okay, melt the helix.
2. Annealing. Cool it down, maybe 50-65 degrees C, to allow short synthetic DNA strands called primers to bind, or anneal, to complementary sequences on opposite strands, flanking the target region you want to copy.
So the primers define the target sequence.
Exactly. You design them specifically for the gene or region you're interested in. 3. Extension. Raise the temperature slightly, usually 72 degrees, the optimal temperature for a special heat-stable DNA polymerase, like Taq polymerase, to add nucleotides to the 3 front ends of the primers, synthesizing new DNA strands complementary to the template strands.
Taq polymerase, that's the one from hot springs bacteria.
That's the one. Its heat stability is crucial because the denaturation step would destroy normal polymerases.
So each cycle, you melt, anneal primers, and extend.
And each cycle doubles the number of DNA molecules containing the target sequence. So you get exponential amplification. 2 becomes 4, 4 becomes 8, 16, 32, very quickly reaching billions of copies.
Incredible. And it's specific because of the primers.
Highly specific. While PCR itself isn't always perfect for making large amounts of error-free DNA for cloning, Taq polymerase lacks strong proofreading. It's fantastic for quickly generating enough of a specific fragment that you can then clone or use for diagnostics, forensics, sequencing, etc.
Speaking of sequencing,
Yeah.
once you've cloned or amplified a gene, how do you read its actual code?
That's DNA sequencing. Early methods like Sanger sequencing were breakthroughs, but now we have next generation and third generation sequencing technologies that are incredibly fast and cheap by comparison.
What's state of the art now?
There are amazing techniques. Some, like nanopore technology, involve pulling a single long DNA molecule through a microscopic pore, a nanopore. As each base, A, T, C, or G, passes through, it disrupts an electrical current in a unique way, allowing the sequence to be read directly in real time.
Reading DNA like feeding thread through a needle. Wow.
It's truly remarkable progress.
And maybe the most mind-blowing application is actually changing the sequence, gene editing.
Yes. This has moved from science fiction to reality, largely thanks to the CRISPR-Cas9 system. It's completely revolutionized the field.
CRISPR-Cas9, I've heard a lot about it. How does it work?
Cas9 is an enzyme, a nuclease originally from bacteria that cuts double-stranded DNA. Think of it as molecular scissors again.
Like restriction enzymes.
Similar in that it cuts DNA, but different because Cas9 doesn't recognize a fixed DNA sequence on its own. Instead, it's guided to its target by a molecule called a guide RNA.
So the RNA acts like a GPS.
Exactly. The guide RNA is designed to have a sequence complementary to the specific DNA target you want to cut. It binds to that target DNA sequence, and then the Cas9 protein cuts both strands right there.
So you can target any DNA sequence just by changing the guide RNA.
That's the power of it. Incredible precision and flexibility.
What can you do once you've made the cut?
Two main things are incredibly useful. First, you can knock out a gene. If you just provide the Cas9 and guide RNA, the cell's natural repair mechanisms will try to patch the break, but they often do it imperfectly, inserting or deleting a few nucleotides, creating a mutation.
Right, which usually inactivates the gene. This is invaluable for researchers trying to figure out what a gene does. Just disable it and see what happens.
Okay, disabling genes. What's the second use?
Repairing a mutated gene. Here, along with the CRISPR-Cas9 system that cuts the defective gene, scientists also provide a template, a piece of DNA carrying the normal, functional version of the gene sequence. When the cell's repair machinery fixes the break made by Cas9, it can use this provided template to correct the sequence, effectively replacing the faulty gene sequence with the healthy one.
So you can potentially correct genetic diseases at their source.
That's the enormous potential. Researchers are actively working on using CRISPR to treat inherited diseases like sickle cell anemia, cystic fibrosis, Huntington's disease, even exploring approaches for Alzheimer's, Parkinson's, and some cancers.
Have there been successes?
Yes, promising results are emerging, particularly in lab settings and animal models. For example, editing blood stem cells from sickle cell patients ex vivo outside the body and showing they can produce healthy hemoglobin. Clinical trials are underway for several conditions.
Are there still challenges? Concerns?
Definitely. A major concern is off-target effects. The possibility that Cas9 might cut at unintended sites in the genome that are similar, but not identical, to the target sequence. Researchers are working hard to improve specificity and detection methods. And CRISPR isn't just for medicine, right?
No, it's being explored for many things. Altering crop genes for better yield or nutrition. Modifying insects, like mosquitoes, to make them unable to transmit diseases like malaria or Zika. Sometimes using controversial gene drives.
Gene drives, what are those?
It's a way to bias inheritance. Normally, a gene variant has a 50-50 chance of being passed on. A gene drive linked to a CRISPR system can essentially copy itself onto the other chromosome, ensuring it's inherited by almost all offspring, rapidly spreading the trait through a population.
Wow, that sounds powerful. And potentially risky.
Extremely powerful, and it raises significant ecological and ethical concerns. Which brings us to the broader ethical landscape.
Yeah, Jennifer Doudna, one of the co-discoverers, has been quite vocal about the need for caution.
She has. Recognizing the incredible power but also the potential for misuse, she played a key role in convening international summits to discuss the ethical implications, particularly around editing the human germline changes that would be heritable.
Changes passed down through generations.
Exactly. It underscores the profound responsibility that comes with the ability to rewrite the fundamental code of life.
So we've really covered a lot, from those first hints about a transforming principle, through cracking the code of the double helix, understanding the complex dance of replication and repair, how DNA is packed, and now the ability to edit it with CRISPR.
It's an amazing journey. It shows DNA isn't just a static blueprint, it's dynamic. It's copied with incredible fidelity, constantly maintained, precisely packaged, and now, thanks to decades of scientific ingenuity, it's becoming a molecule we can engineer.
So what does this all mean for you, listening right now? It means the very code that defines life is being decoded and manipulated at a pace that's just unprecedented. It's opening doors we couldn't have imagined just a few decades ago.
And for our final thought, building on that ethical dimension, given these powerful gene editing technologies like CRISPR-Cas9 and the speed at which they're advancing, how do you think society should navigate this? How do we balance the incredible potential benefits curing genetic diseases, fighting pandemics, with the profound ethical responsibilities of altering the very basis of life, potentially in ways that affect future generations?
That's definitely something to reflect on, a crucial conversation for all of us. Thank you for joining us on this deep dive. We hope you've gained a clear understanding of the molecular basis of inheritance.